So we talked about the adaptive immune system. We talked about B cells and T cells, both helper and cytotoxic, and what they do. Now I want to talk about how they come to be. So I told you that B cells are called B cells because they mature in the bone marrow, and T cells are called T cells because they mature in the thymus. And actually both of them start out in the bone marrow, but T cells migrate to the thymus. But once upon a time, we didn't have bone marrow. When we were zygotes and maturing embryos, we didn't have bones. So where the heck did all this hematopoiesis that makes the B and T cells start out? Well, it starts out in this place called the yolk sac, which is part of the embryo. And then as we grow into a fetus, it's going to migrate into the liver and spleen. And when we get to be a kid, it's going to be in what we call our appendicular skeleton. So this is going to be like our arms and our legs. And as we grow, it goes into our axial skeleton bone marrow, and that's going to be like our middle or our trunk. So that's where hematopoiesis occurs, but how do these B cells and T cells get so much diversity that they can bind specifically to basically any pathogen that just happens along our bodies? Well, this process is actually pretty cool. There is a protein complex called recombinase, and it's made from these genes called RAG1 and RAG2, and this recombinase will go into the DNA, and it will actually cut it up and rearrange it, and there's this other enzyme called terminal deoxyribonucleotide transferase, or TDT, and TDT puts like little random sequences of nucleotides in, and then the mismatch repair system, which is what helps us correct mistakes in our DNA, is going to fill in the other side with complementary bases, because TDT only makes single-stranded stuff. But anyways, this is going to produce a lot of diversity in the sequence, and even more diversity will be created through RNA splicing. So remember that RNA splicing is something that happens after we do transcription to make RNA, and it's going to be done by this protein and RNA complex called the spliceosome, and all the introns are going to go away and get degraded, and all the exons are going to stay. And so alternative splicing is when we change up which ones are the introns and which ones are the exons, and that's what happens in order to make this incredible diversity of variable regions that we have. And it's very interesting because clinically this can go wrong. There's something called Omen syndrome, and Omen syndrome is when you have missense or just changes in mutations in the RAG1 and RAG2 genes, which remember make the recombinases that switch around the DNA, and this results in a total lack of B cells. And people with this syndrome have a lot of immune deficiency. They'll have rashes, they'll have diarrhea, they'll have a lot of clinical problems. There's also this thing called severe combined immunodeficiency, and this is where you have null mutations, or mutations that just render the proteins completely non-functional for RAG1 and RAG2 genes. And so this recombinase basically doesn't exist in these people, and they don't get this kind of diversity, and this basically crashes their adaptive immune systems, and they have a total immune deficiency of both B and T cells. So this recombination is really, really important. But anyway, once we've done this recombination, we encounter some interesting hurdles here. So when our B cells are in the bone marrow and they're maturing, some of them, just by chance, because we're making so many combinations, on the order of billions, that some of them are going to bind to things that are in us, self-antigens. And this can cause not immunodeficiency, it can cause autoimmune disorders. So for example, diabetes mellitus type 1, that's going to be where your immune cells attack your own pancreatic beta cells that make your insulin, and then you can't regulate your blood sugar anymore. So the body tries to avoid outcomes like this by selecting against the B cells and the T cells, but right now we're talking about B cells. It's going to select against the B cells that do this, that have an autoimmune response. And the way that this works, there's two mechanisms. One of them is called deletion, and this happens in the bone marrow. And that's basically these cells will die. And then there's also something called anergy, and this happens once they get out of the bone marrow. And this is going to be, basically they're going to change their composition of the antibodies on their surface so that they are rendered inactive. So I told you that the variable region changes up and is subject to all this recombination stuff. But what about the conserved region? Well, the conserved region has like this set of things that it can be. And this is what people refer to when they're talking about, say, like IgD or IgE or IgM. Ig is just immunoglobulin, which is another word for antibody. But these cells are going to express a particular IgD isotype of antibody. Quick vocabulary thing, isotype is going to be referring to these letters, like the set options for the conserved region, and idiotype is going to refer to the different types of variable region, which is like every single B cell has a different idiotype, but all the B cells might share a set of isotypes. But anyways, anergy is going to involve an overexpression of the IgD isotype. So the variable region is still going to be problematic. It's still going to be autoimmune and bad. But the fact that all the conserved regions are IgD type is going to set off some type of signal that renders these cells inactive because they have too many IgDs. The T cells, meanwhile, once they are ready to mature, they're going to migrate from the bone marrow to this organ called the thymus. And the thymus is this little immune organ near your heart, and it's got a cortex and a medulla, and you might recognize this from, say, the kidney or the brain. And the cortex is just the outside, and the medulla is just the middle. And these T cells are going to start out in the cortex, and as they mature, they're going to head over toward the medulla. And when they first come in, these T cells, they don't know what they want to be when they grow up. They don't know if they want to be helper T cells or cytotoxic T cells. So at first, these T cells are going to express CD4 and CD8. Remember that helper T cells express CD4 and cytotoxic T cells express CD8. So these immature T cells are going to express both CD4 and CD8. They're also going to express, of course, their T cell receptor. I'm not going to write that down because that's pretty much implied for all of them that they have a T cell receptor. That's not going to change throughout the steps that I'm about to walk you through. And then in these early stages, these cells are also going to express another thing called CD3, and that's going to kind of just let us know that they're still immature. And what's going to happen with this is we call it double positive, because they have CD4 and CD8. They don't know what they want to be yet. Then they're going to go through some selection. So in this next step, they're going to encounter some self-antigens, just like the B cells did. And these self-antigens are going to be presented by some professional antigen-presenting cells and some regular old cells with an MHC1 that are hanging out in the thymus. So we've got some dendritic cells, we've got some macrophages, and these guys are going to present with MHC2 complexes, self-antigens. And there's also some epithelial cells, which are just like regular old run-of-the-mill cells, so they have an MHC1, and they're also going to express self-antigens. So this is kind of like a test or a gauntlet for the T cells to go through to make sure that they are the right ones. And the right ones, they are very rare. This is a very rigorous process. About 95 to 99% of T cells are not going to make it through this. They're going to die. So this is like the Hunger Games of immunity. But anyways, so we want these T cells to bind to the MHC complex, because that's what they're going to have to do in the future. They're going to have to bind to antigens being presented by the MHC complex. But we don't want them to have affinity for these self-antigens, or else they're going to cause autoimmune problems, right? So if these cells have high affinity, that means that they like the MHC complex, which is good, but they also like the antigen, which is bad. So these cells are going to undergo negative selection. And that is going to tell them that they need to apoptose and die. If these cells have no affinity at all, so they don't bind to the MHC complexes, so this is all referring, these high affinity and low affinity things I'm writing are referring to the MHC complex presenting a self-antigen. If there's no affinity, that means they don't like the self-antigen, which is good, but they also don't like the MHC complex, which is very problematic. So these cells, they're not as bad as the ones that like the self-antigen, so they're not going to get negatively selected, but they're also not going to get positively selected. So they're just going to kind of stay where they are, and they're not really going to grow up anymore, so eventually they're going to die. So this is like nothing. And if you have low affinity, that means that you like the MHC complex, but not the antigen. So that's good, right? Because we want them to be attracted to the MHC, but we don't want them to be attracted to the self-antigen. So these ones are actually going to get positively selected. What am I doing? This won't let me write selection. So they're going to get positive selection, and positive selection is going to tell them grow and divide and differentiate some more and go to the thymus medulla. The last thing that's going to happen is that these cells are going to decide whether they want to be helper T cells or cytotoxic T cells. So if they bind with higher affinity to MHC2, think about what kind of cell they might turn into. If they're binding MHC2, which is expressed by these professional antigen-presenting cells like dendritic cells and macrophages. Pause the video if you want to take a second to think. Okay, so these guys are going to be helper T cells, so they want to have CD4. So these guys are going to be single positive. And if you bind MHC1, that means they're binding the complex that's on all the nucleated cells in the body, the basic one. So if a cell gets infected, they present an antigen on MHC1, and then they need to die. So if these T cells bind MHC1, they're going to be cytotoxic T cells, which means that they are going to they're going to express CD8. So these guys are termed single positive. And that's how we get T cells. And so from the bone marrow and from the thymus, these B and T cells are going to navigate their way into some other immune tissues or organs. So we talked last video about the lymph nodes and how all the tissue, the interstitial fluid gets filtered through the lymphatic vessels, which are eventually going to take this fluid back into the blood. But before the fluid gets back into the blood, it's going to pass by these things called lymph nodes. And these lymph nodes have a bunch of immune cells, which are going to detect any pathogens and start an immune response. And they're going to chew up the pathogens and kill them and not let them into the blood, which is where this fluid is ultimately going, right? So there's the lymph nodes. This green thing is the structure of a lymph node. I wouldn't worry too much about the structure. This comes from a USMLE prep book, so yeah, do not worry about it too much. But these guys are called follicles. They're groups of cells, and they're going to contain a lot of B cells. This one's got a cortex and a medulla, just like the thymus does. And then the spleen, for such a long time, I knew what almost every organ did but the spleen. The spleen was like this mysterious thing that I didn't know what it did. But what the spleen does is it's got some immune cells from the adaptive immune system. It's got B cells, it's got T cells, and the spleen interacts with the blood. So the lymph node interacts with the lymph, which is connected to like all the tissues and the interstitium, right? But the spleen interacts with the blood, and because the blood is not very permeable to pathogens, if you have a tissue infection, you're not going to have access to the blood immune cells, so that's why you need the lymph node. But if you have a blood-borne pathogen, then you need something else, because that pathogen's not going to make it into the tissues and into the lymph. It's just going to hang out in the blood and cause a bunch of chaos. So that pathogen is going to pass through the spleen, and the spleen is going to launch an immune response to that pathogen. So here's a little summary of what goes on in the immune response with these immune organs and these immune cells. So if we have a tissue and it gets infected, then if it's in the blood, it's going to go to the spleen. That's this little part. And if it's in the tissues, it's going to go through the lymphatic vessels, which kind of have like a closed off end at the tissues, but it's very permeable, so it's going to let fluid and pathogens in through it, and it's going to go toward the lymph node. And in the lymph node, the immune cells, the various immune cells, are going to get activated. So this is activation. And then these are going to be able to go and launch their response. So that's how B cells and T cells come to be. And I thought this topic was super interesting, so I went a bit in depth. If you're studying for high school or college biology class, even if you're prepping for the MCAT, you don't have to know all the details about RAG 1 and RAG 2, but I thought that the clinical application was kind of cool, so I went into it. I hope this video was as fun for you as it was for me.